video lecture is going to look at male reproductive system, looking at their anatomy, the process of making sperm called spermatogenesis, and then finally how male hormones are regulated. So starting with the anatomy, of course, starting with the testes, this is the location of sperm production. The testes is housed inside of a sac called the scrotum. Notice also the testes then is outside the body cavity to reduce temperature in order to maximize sperm production. The sperm actually complete their development in the epididymis, and they are stored in that epididymis until time of ejaculation. The sperm will then travel up the ductus deferens and enter the body cavity, traveling still in the ductus deferens until it joins with the ejaculatory duct. The ejaculatory duct then combines or becomes the urethra, where the sperm then are delivered to the female through the penis. The glands that add to the semen, these are the seminal vesicles, the prostate gland, and the bulbourethral glands. Now, sperm, again, are produced inside the testes in what are called seminiferous tubules, which is what composes most of the testes. And you can see here a seminiferous tubule in cross section. The seminiferous tubules contain what are called spermatogonia. These are the stem cells that will end up producing the sperm. And then the other cells are the sustenticular cells. These cells are supporting cells that play a role in sperm production, which we'll see in a little bit. And then finally, outside of the seminiferous tubules are cells called the interstitial cells or Leydig cells, and these cells produce the androgens, mostly testosterone. In spermatogenesis, we're going to again produce sperm, and the description for this is on the next slide, so you'll want to follow along on that slide. We're going to start with the spermatogonium. The spermatogonium, again, is the stem cell, and notice he's diploid, so he has all 46 chromosomes. It undergoes the process of mitosis to produce two daughter cells. One of those daughter cells is called a type A cell that remains a spermatogonium, whereas the other daughter cell is a type B cell, and that one will continue to grow and develop and become what's called the primary spermatocyte. Now the primary spermatocyte, though, notice is still diploid. This primary spermatocyte will undergo meiosis, the first meiotic division, and then notice it's a reduction in the number of chromosomes in that first meiotic division, so now it's a haploid cell called the secondary spermatocyte. The secondary spermatocyte then undergoes meiosis 2, again dividing, but since it's already haploid, those cells remain haploid, and those are spermatids. The spermatids then in turn undergo a process called spermiogenesis that changes the shape of the spermatid into our spermatozoa, or it's just simply sperm. And so here you can see kind of the process going from the spermatogonium to the primary spermatocyte, the secondary spermatocytes, the early spermatids, and finally our sperm once it enters the lumen of the seminiferous tubules. Now, to help with this process are the sustenticular cells. We can see here the cytoplasm shown in green, and then the purple is the nucleus of the sustenticular cells. And the sustenticular cells have several different roles. One is to provide nutrients to the developing sperm cells, also to help move the cells along so that as you continue from one stage to the next, it's pushing the cells towards the lumen of the seminiferous tubules. It also secretes a rich fluid in androgens or testosterone to help maximize sperm production. It will phagocytize faulty germ cells. And then last, it produces inhibin and androgen binding proteins that help in regulating spermatogenesis. Now, spermiogenesis is the process where we transform the spermatid into an actual sperm, or spermatozoan, same thing. In this process, I want you to notice that the cytoplasm is significantly reduced down to pretty much nothing, so that all you have left is here's this little teeny bit of cytoplasm on the side. So the final sperm, you have what's called an acrosome. The acrosome has a lysosome-like structure that contains hydrolytic enzymes that are used to penetrate and enter the egg. And then, of course, we have the mitochondria. The mitochondria are housed in what's left of the cytoplasm, and those mitochondria, being the powerhouses, are going to help in that tail to work, give the energy source for the tail to work. The nucleus, of course, is where we're going to find the 23 chromosomes. And then last, of course, the tail is also called the flagellum. Now, male sterility or low sperm counts can be due to several things. One are called xenobiotics. These are substances in our body, but not from natural sources. So these could be environmental toxins, phthalates, which are associated with plastics. That phthalates are in plastics to help make them more flexible, but those have been linked to low sperm count. Hormone-injected beef also may cause problems with male sterility because it contains a high amount of estrogen-like compounds, and that estrogen then can end up blocking the action of the male sex hormones and therefore reduce sperm counts. There's antibiotics used by the meat industry has also been linked to suppressed sperm formation. Radiation, lead, marijuana, excessive alcohol can also cause abnormal sperm formation. And then, of course, the age-old question of boxers versus briefs. That is the idea that since the sperm need a lower than normal body temperature, wearing briefs would pull the scrotum and testes up close to the body and therefore warm it too much, whereas boxers wouldn't do that. The male duct system, as I mentioned before, includes the ductus deferens, the ejaculatory duct, the urethra, and along the way as the sperm travel, things are added to it through the glands. The 3 glands are the seminal vesicles. These seminal vesicles produce 70% of the semen by volume. It contains things like fructose for nutrients. Its alkaline fluid neutralizes the acidity of the vagina. It also has coagulation enzymes that keep the semen stuck to the vaginal wall so they aren't eliminated. It has prostaglandins to decrease the viscosity of the mucus in the cervix to therefore allow the sperm to get through that cervix and through the mucus in the cervix. And finally, it has a yellow pigment in it that fluoresces under UV light. Now that doesn't help or has no function whatsoever in male reproductive system, but since it does fluoresce under UV light, it can be used as a way to find semen in a crime investigation. The prostate gland contains fibrinolysin. This fibrinolysin makes the semen less viscous, so the sperm can swim into the uterus. I mean, once they've been stuck in the wall, then it'll become activated later so that they can swim once they're entering into that uterus. And also plays a role in activating the sperm. And then finally, the bulbourethral gland contains a mucus secretion that lubricates the penis during intercourse and then can neutralize the acidic urine contained within the urethra. Typically, a male will ejaculate 2 to 5 milliliters by volume, but that may not seem like a whole lot, but that will actually contain 120 to 150 million sperm per ejaculate. Now, to regulate hormones of the male reproductive system, we're going to start with the hypothalamus. Now the hypothalamus produces GnRH, or gonadotropin-releasing hormone. This gonadotropin-releasing hormone then, here, stimulates the pituitary gland to release FSH and LH. Now the FSH then stimulates spermatogenesis, and it does this by getting the sustentacular cells down here in that testes to release androgen-binding proteins. Now these androgen-binding proteins keep testosterone levels high surrounding the spermatogenic cells, and that's what's going to allow having all that testosterone around those spermatogenic cells to end up allowing spermatogenesis to continue. LH then stimulates the interstitial cells. Remember, the interstitial cells are in charge of producing testosterone. Now, testosterone does a number of things. One, it does stimulate spermatogenesis. So more testosterone, the more sperm you're going to make. But it also then stimulates the secondary sex organs, as well as stimulates the secondary sex characteristics, particularly at puberty, and then continues to enhance those secondary sex characteristics through the rest of life. So it's going to increase metabolism, muscle growth, and erythropoiesis, or red blood cell production. And then finally, testosterone will also increase libido, or sex drive. To regulate levels, testosterone inhibits GnRH. So if I inhibit GnRH and FSH and LH, that means high levels of testosterone means low levels of GnRH, and therefore FSH and LH. If I have less LH, that means I make less testosterone, less inhibition, which means more GnRH, more FSH and LH, which means more testosterone, and again, more inhibition. So it's kind of a negative feedback system to keep testosterone levels pretty constant. Inhibin is also produced by the sustentacular cells, but it's produced in response to sperm counts. So as the sperm production increases and I get higher and higher sperm counts, more inhibin is released. Inhibin then in turn inhibits FSH and GnRH when sperm counts are high. So the more inhibin I have, the more inhibition I have of FSH and GnRH, which means then less ABPs, less of those antigen binding proteins, which means testosterone levels in the testes would be lower, and therefore spermatogenesis would be lower, and my sperm counts would be going back down. And then once those counts go down, I would mean less inhibin, less inhibition of GnRH and FSH. More FSH means more antigen binding proteins, which means higher testosterone levels in the testes, which means increase in spermatogenesis. And again, a negative feedback system to help maintain testosterone, or maintain sperm production and testosterone levels. And so those levels are pretty stable. So that's our look at male reproduction. The next video lecture, we're going to look at female anatomy and reproduction.